Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Mike Burman, who's an associate professor at the University of New England. I've known Mike for years and have always been impressed with his passion for communicating science to the public. Not only did I learn a lot about Mike's work in our conversation, I think his reasons for doing the work he does is a great representation of most of the scientists I've met, the desire to make the world a better place. Mike was a 5-minute genius speaker at the 2018 Maine Science Festival and is an advisor to the Maine Science Festival. I've included links to Mike's UNE webpage and his 5-minute genius presentation in the show notes. Welcome to the Maine Science Podcast.
Yeah, thanks, Kate. Thanks so much for having me. I'm an associate professor of psychology. I'm in the School of Social and Behavioral Sciences. We're trying to consolidate our programs a little bit, give faculty more access to each other and to collaborations. We're also trying to simplify the path for students. So students can come through with bigger programs, making it more seamless to take courses across disciplines.
And how did you end up at UNE?
I'm actually originally from the Midwest. I grew up in Wisconsin, and I did most of my schooling in Minnesota. But I married a Mainer. We met in college. And so we had a child, and having a child not near family wasn't so much fun. So I started looking for jobs close to my family or hers, and ended up in Maine. And UNE is a great fit. They have a big neuroscience program, a bunch of people I can collaborate with and work with. And I was lucky. It wasn't so planned as much as it was just fortune, but it was good fortune. It's worked out really well.
It's hard to plan with academics. You rarely get your pick of the litter.
Well, that's absolutely true. I also think life is like that, right? You take the opportunities you have, and looking back, it kind of makes sense. But looking forward, it's hard to predict.
Was your undergraduate work in neuroscience as well? Isn't that that would be an unusual major for an undergraduate degree, right?
Less unusual now. UNE offers a neuroscience major, as do lots of other programs. I'm actually a member of a group called the Faculty for Undergraduate Neuroscience, and they try and spread programs around. But when I was an undergraduate, it would have been really rare. Neuroscience was still a blossoming discipline. I got my bachelor's degree in psychology at a little college called Carleton College. It's in Northfield, Minnesota. Northfield, Minnesota has the slogan of cows, colleges, and contentment. And it's just kind of one main street and two colleges. So I went to one of them. It's also where I met my wife, by coincidence. But no, I did my bachelor's degree in psychology. I fell in love with the psychology of learning. It's called, kind of classical operant conditioning, Skinner, Pavlov, that sort of thing. Although it's a tiny, tiny college, about 2,000 people, they had a primate colony there. We had cotton-top tamarins, which is really rare for a school that size. And my mentor, Julie Nyworth, was just a powerhouse, still is a powerhouse in science and as an instructor, and really just turned me on to science and to psychology and to the psychology of learning. So I did a senior thesis with these monkeys, trying to understand about their memory and foraging and cognition and how they found food and kind of adapted to changes. And we also published a paper on some social communication and how they did that.
Were you surprised by any of your work with them?
Well, it's funny. As an undergraduate, I was really naive. And it was all an accident. And I listen to some of your podcasts and I hear different science people's trajectories. And it seems like some scientists really set out to do their life's work. And I wasn't like that. I was actually playing poker one night with somebody. And they said, Mike, you're really smart and you're a psychology major. You'd be a really good fit for our primate lab. I'm like, that sounds great. I'll sign up tomorrow. And that's how I ended up in science, just purely by coincidence, just meeting the right person at the right time who chatting about my interests and what I wanted to do, pointed me in the right direction. Thanks, Ricky, if you ever hear this. So I was just fascinated. I was fascinated by memory. I was fascinated by what turns out to be memories that are dependent on a brain structure called the hippocampus. But I didn't know that at the time. And so my undergraduate thesis was trying to understand how monkeys found food. And so just a really naive question. You're all going to laugh, anybody listens to this. But I just took their big enclosure and I set up 2 kind of foraging boxes where the monkeys could go into one or the other to find food. And I put really discrete cues on each one. So I put some particular shapes and colors on each one. So they were different until they got used to finding food in one and not the other. And each set of monkeys had a different arrangement of where they could find their food. And then I just switched them. I thought I wanted to find out if they went to the same location or if they followed those cues. And it turned out that exactly half the monkeys took each strategy. And so it was completely inconclusive. And I didn't know, whatever. That's what I found. That's what the monkeys did. But so, I hope to really answer this question and really solve this thing. And it turned out I just didn't really, there was not much to learn from the way I organized it.
That's also really important to learn, right? Maybe this wasn't a great design, or maybe it just wasn't the right set of circumstances.
That's right. Or there's not a predictable way to tell with the monkeys. And, it was fine. I worked really hard on it. I wrote it up well. I put it in the context of literature, and it was a really good experience, even if I didn't quite have the outcome I wanted. We also published a paper on whether monkeys could follow human cues. And that was kind of the work that's now been published and cited more. And kind of a similar design. We hid food under like a Dixie cup at the time, I think. And there were multiple Dixie cups. And then the human experimenter would either kind of tilt their body towards it or look at it or point to the correct cup to see if the monkeys could follow those social cues. And this was actually a replication of some other work that had come into question. There was a researcher who had published a lot of cognition work on cotton-top tamarins. Then it turned out that some of their research wasn't conducted maybe as thoroughly as it should have been. And so we were replicating some of their work to understand if it held up. And this one did. They were able to follow human experimenter cues in order to find the right food. So that was interesting also. And it's kind of fun to be in a cage and kind of communicating with these. They're just little 1 kilogram, 2.5 pound monkeys. They're cute. They're kind of fun. So it's fun to be in there with them and communicating with them and understanding about these, social cues and how they seem to be then primate general, right? Just communicate with each other. That's a broader trait. That's where I started. Really developing the science and the science process, and especially again, this science of learning and behavior change.
So did you stick with primates for graduate work, or did you do a whole other different?
No. Much to the dismay of my undergraduate mentor. I think Julie's probably still mad. I did not stick in primates. Again, my career is the story of meandering. And what I knew as I finished my undergraduate education is that I loved this science of learning. I was really fascinated by learning and memory and how they shaped us and how they shaped our experiences and how they shaped our personalities. And so I applied to a host of programs. I applied to developmental programs where people were studying memory in infants and development. I applied to other primate labs. I applied to behavioral neuroscience labs where people were studying the neurobiology of memory. And that's what I ended up doing. I ended up going to the University of Minnesota, somebody called Jonathan Gortz, who was studying the neurobiology of aversive memories, of how we encode trauma, really. And he was sort of interested in a brain structure called the amygdala and what happened in the amygdala. And I was interested in this brain structure called the hippocampus and how it interacted with the amygdala. And so I did my PhD on how the hippocampus and amygdala kind of cooperate during the formation of traumatic memories. And we used rats. We used a rat model for those experiments. And that's where I learned my neuroscience. I actually originally wanted to be a physician. I went to college thinking I wanted to be a medical doctor. I did not like my biology classes as much as my psychology classes. So I left biology, went into psychology, and then kind of came back around to biology once I understood how useful it was for understanding these questions.
Was it more interesting to take biology once you had an end goal of the brain?
Yeah, I think that's exactly right. So, I'm motivated by my questions and kind of my passions and what's interesting to me. And it turns out that I'm interested in using the tools that biology offers to answer these psychological questions. I want to understand memory. I want to understand emotions. I want to understand the mind. I want to understand how humans work. So those are really psychology questions. But it turns out some of the right tools to answer those questions have to do with biology, understanding the brain and circuits and cells and molecules and receptors. That's what makes neuroscience fun. It allows us to kind of tie together those disciplines and use those tools.
How translatable is the work that you did with rats to the human brain? I mean, I'm assuming people pick these models for both ease of study, but also because you can make strong conjecture or hypothesis.
That's a really good question, Kate. So, it's interesting. I'm going to tell you a little bit of a long story, and then we'll come back to the direct answer. So if you're interested in emotions, there's kind of two ways people go right now. They study kind of either positive emotions like happiness, joy, resilience, or they study negative emotions. And there's been a real push recently, this movement called positive psychology to study positive emotions, happiness, joy, contentedness. But one of the difficulties there in studying positive emotions is that they're really hard to define, right? When I say I'm happy and you say you're happy, it's hard to know if we mean the same thing. I mean, maybe I'm talking about the large pepperoni pizza in front of me making me happy, and maybe you're talking about really succeeding in your life's goals, or making some progress towards that. And we use the word happiness for both of those things, but they're really not the same at all. And, you might think the same thing is true of drugs of abuse, right? Do they make you happy? I mean, they make you feel better in some sense, but probably not happy in the same way as, as doing really well on a long-term project. And probably not in the same way as hugging a close friend or touching your lover's hand or whatever these things are that feel good. And so we kind of think about that. And then you think about how to study the neurobiology of those things, how to create animal models of those things. And that really makes it complex, right? I don't know what you mean by happy. I certainly don't know what makes a rat happy. And so trying to understand that neurobiology and whether even or not it's conserved becomes a really hard question. And the things that we do know is some circuits are conserved, but many others, the ones that we probably really mean when we say happiness or content or those things, we're not even exactly sure what they are in humans, much less knowing if rats have those emotions or those brain structures or how homologous they are to us. That same thing is not true of negative emotions like fear and threat processing and anxiety. There it's pretty clear. If I tell you that you're going to experience an electric shock, I know exactly how you feel. We feel about the same about that. And we engage in the same behaviors, right? We sweat, our heart rate goes up. If there's a threat out there, if we hear a loud, sudden sound, we both kind of freeze and orient towards it. And not only that, but we actually share those behaviors with almost all other vertebrates and a lot of invertebrates too, right? You freeze in the headlights just like a deer does or a rat does. It's no different. And it turns out that circuitry is really well conserved, right? In part because we have these same reactions and these same responses, right? A rat also doesn't like being shocked. A rat will also freeze when it thinks a shock is coming. Its amygdala will be activated. It has very homologous, not only that sort of superstructure of the amygdala, but the subnuclei too. The neurotransmitters are very similar. And so there we can really study the neurobiology of these negative emotions really well in a way you just couldn't with positive emotions. So when we ask, do rats experience emotions like humans? It depends on the emotion. So when we look at threat processing, we can really learn a lot in ways that we couldn't from humans and wouldn't from humans, right? We wouldn't, these experiences would be unethical.
Is it because of evolution and that part of the brain, the amygdala and is it hippocampus? Are they the oldest parts of our brain, so to speak, kind of evolutionarily speaking?
Yeah. So that's depends on how far back in evolution you want to go. When we think about evolution and kind of the phylogeny of the brain, we actually started in the brain stem. We think that was kind of the first structure to emerge, and you'll see that in amphibians and fish and all vertebrates kind of share those basic structures. There's not a lot of emotional or motivational structures there. There's some very primitive ones. There's also some primitive kind of sensory motor structures. And then as we get kind of one step higher, we get into our midbrain that kind of sits on top of our brainstem. And again, that's where we start to see some of our basic reward pathways, some of the ones that are involved in, say, drug addiction. And then we start to get into what we call our forebrain, which is a little bit more evolutionarily advanced. And some of the older structures in our forebrain then do include our limbic system, which is our amygdala and hippocampus. And resting on top of that, we have our neocortex, which is kind of the things that we think become more primate specific and maybe a little bit human specific. And then we get into some parts of cortex, our really frontal cortex, maybe even kind of more human specific or have evolved more recently. So it depends on how far back you want to go, Kate.
When did you neuroscientists, figure all that out? Is this relatively new in the world of science?
So these are some of that work called the triune brain. Paul MacLean did it. This is kind of 1960s era science. So new for science, not new for neuroscience. Neuroscience, moves rapidly.
You're still working on the negative emotions, right? Your research now. Maybe you can tell us. I don't want to presume anything else other than that. So if you can dive into what it is you're doing now, and kind of how you do it. Right, because like you said, it seems like, I think it seems very hard and difficult to study the brain for a whole host of reasons. So maybe you can kind of tell us how you structure it so you actually know what's going on.
So I decided, as I was kind of finishing my PhD, I decided it was really important that my science was practical and that not just for my interest, but I thought could serve some societal good. We take a lot of taxpayer dollars. And we ask rats to both suffer and give their lives for our research, and I think those are real ethical concerns and a real place, a real ethical burden, I think, on me and our research. And so it was important to me to believe, and I still believe, that our research be making an impact. We should be making human lives better if we're going to do these things. And so we picked a problem that I thought was a significant problem and one that I thought we could make an impact on. And the original problem we tried to address was anxiety and the neurobiology of anxiety. And when I started this line of research now about 13 years ago, We were kind of in this decade called the decade of the brain. President Obama had kind of named it. And we were learning so much about the brain and about how it processed emotions, including anxiety. And people were starting to draw circuit diagrams of what cells were involved and what neurotransmitters were involved. But I really thought I identified a hole in all of that. And as awesome as that work was, and as meaningful as it was, and as much progress as that research made, nobody was studying it from a developmental standpoint. All that work was being done in adult organisms with the goal of creating anti-anxiety medications that would treat adults with anxiety. But as you may know, and as some of your listeners may know, anxiety really emerges early in life. Specific phobias tend to emerge around 6 years old. Separation anxiety around the same age. But even generalized anxiety tends to emerge in early adolescence, around the time of puberty. And I really thought that we were missing the boat in not trying to understand the development of those circuits and when they were vulnerable, what kind of changes or what kind of events would cause changes in them that might predispose one towards anxiety. And really starting to take some of this knowledge we had about the circuits and neurotransmitters and brain structures and really understand how that came to be and how those changes came to be and where we could intervene and where those vulnerable periods were. And so that's kind of what I set out to do, is to understand the development of anxiety and fear, with kind of the idea that once we understood normal development, we could then understand abnormal development and how that occurred in people with anxiety disorders, and then hopefully, of course, eventually intervene. Now, it turns out, I think the world gets ready for ideas. And so although I think I had the idea early, I wasn't alone, and many other people did around the same time. And NIH had some funding set aside to answer these questions, which I was, fortunately benefited from, but so did others. And so we've learned a lot in the ensuing decade. And we published some work, and many others did. And we really now understand the developments of anxiety and fear and how it compares to adults and different ages, which different functions and brain structures come online. And really did a lot of that work. And I think the situation has really changed. And although there's still a lot more to do, we're no longer completely ignorant of that. And so we've kind of then moved on to the second phase. And we become interested in what can perturb the development of those systems. I had a personal experience around my daughter's birth. She ended up back in the NICU on her second day of life. And I sort of watched kind of life in the NICU for her. And it was an incredibly traumatic time for her, for me, for my wife, for her whole family. And I have to say that the NICU saves lives. Kids survive now because of the medical intervention we can do. We can keep children alive who would not otherwise be alive. And that's, you can't speak highly enough of that. That's very important. But it's also an incredibly stressful place. It's loud, it's bright, it's noisy, it's painful, it's disruptive. Babies are living in incubators and they're being handled by strangers and nurses, and they're having blood drawn and pricked and poked and injected. And at the time, and still even a little bit now, there's still complete misunderstanding about what the developing nervous system can process and how that pain works and what the long-term consequences of those kinds of experiences are. And so we know from humans that kids who spend time in the NICU are much more likely to suffer from things like anxiety, fear, addiction, chronic pain later in life. And so we become interested in that and how early experiences shape the nervous system and what those consequences are. And again, with the eye of eventually creating interventions and treatments. And there, I think we've made a real impact. I think we've been testing hypotheses that nobody else has had. I think we've developed our own really niche in the field where we've been studying and identifying changes in the brain uniquely. And I think we've really added to the literature in that way. It's been really, in that sense, really gratifying. I think we're on the right path.
So is that changes in the brain based on different ways of working in the NICU or approaching the kids who are in there?
Yeah, so again, we're working with purely animal models right now. We treat baby rats like you might in the NICU. And we actually, it's really fascinating. One of the things that happens in the NICU is that they draw blood from babies' heels. Their veins are too small to put an IV in or to draw blood. And so they do what's called a heel lance. They take a little razor blade like thing, and they nick the heel and they can then draw blood off the skin. And that can happen many times a day for an infant in the NICU, depending on, what the doctors need. And so we just do that. We can mimic that in our rats. We can just take newborn rats. We can take them away from mom. We just take a little sterilized needle. We just poke their paw till just one little drop of blood comes. We wipe the blood off. We make sure it stops bleeding. We put them back in with mom. And we just thought that would kind of be like what doctors do, to these babies in the NICU. We didn't develop this. We borrowed it from another group who was publishing on it. And the The thing that we discovered amazingly to us was that these rats actually seem more resilient. Later in life, they showed less fear and anxiety. The ones that got poked, were actually more resilient later. And it turned out that was just our ignorance. Once we started getting into the literature, there was a little bit of a literature on this already. Somebody had discovered it way back in 1960, and somebody else had discovered it in 1970, and we just didn't know. They didn't use that exact same model. They did some different kinds of stressors. But this was a known phenomenon that we just didn't know about. And it turns out that when you do what we did, where you take the pups from the dam and you poke their paw and you put them right back, what happens is that the mothers engage in a bout of maternal behavior. They lick and groom and calm the pups. And when you allow them to do that, the pups experience a little bit of stress, they then recover, and that actually trains them about how to deal with moderate stressors. That actually gives them a little bit of leg up. They've had a little trauma, they've learned some techniques for coping with it, and they might even be a little bit better off for that. In humans, that's known too. We actually call that kangaroo care. It's an emerging technique for dealing with infants in the NICU. You give them some skin-to-skin contact with mom, you take them off, do the heel lance, put them back skin-to-skin contact with mom, and that actually brings their stress levels right back down, and they seem to be a little bit healthier afterwards. So, we're rediscovering.
Would that work with dads too? Like if it was, if they were both there and let's say the mom is off getting a different procedure, do you think the same thing would work out, or does it have to be maternal?
That's interesting. I do think the same thing works out. In rats, you can't do it because the dad's not around. And in humans, sadly, dads are understudied. As a very involved dad myself, I feel a little bit slighted by that. But I do think it would work.
Yeah, my son, when he was born, he didn't go in the NICU, but they had to do the razor thing. And it was awful. It was just, and I couldn't even watch it. It was just so devastating to me. And I know after every time, my husband was the one who took him because he could be right there with them. I think it's really interesting because I think dads probably can have a similar, if they're allowed the opportunity to have the same effect, it would be really interesting to see if they would. I can't imagine that they wouldn't.
Yeah, there's actually some interesting physiology too. And I don't know if the dads have been studied, but actually, when infants are laying on the mother's chest, the mother's blood dilates a little bit, letting warmth out. There's a whole physiological response that occurs between the infant and the mother, allowing heat to transfer and some pheromones to transfer. And I don't know that anyone has studied that with dads, although I presume it's similar, but it'd be interesting to find out.
Are there any animal models where the dads stick around?
There are species where there's more co-parenting, not ones in popular lab models, as far as I know, although maybe somebody can write in and correct me if they know of one.
Mike, this might be the only time that women are studied more than men, ever.
That's true, yeah. That's true. I can speak more about that in a minute. We find some really interesting sex differences. It turns out you can reverse that whole situation. You can make the rat pups more vulnerable to fear and anxiety later if you don't put them back with mom. If you keep them separate between the pain episodes, like in an incubator, like they do for some infants in the NICU, now you actually make them more vulnerable later. If they don't have that chance for licking and grooming and calming down by the mother, then their stress, levels stay high, and then now you're kind of setting them up for some long-term risk. We also find that even in these pups that had the pain and went back with mom, if they experience a second trauma later in life, so now they get exposed to some sort of traumatic event, now we see that vulnerability reemerge. Now they show kind of chronic pain and lowered pain thresholds and other evidence of susceptibility. So, it's kind of a two-hit process. If you have just the one trauma early, that can actually be a little bit beneficial. But if you experience two traumas, one early in development and one later, now you're vulnerable again.
Are the traumas different? Like the second trauma could be something, not a replication of the first, but another type of physical trauma.
Yeah, so we've done a couple of things now. We've done something called fear conditioning, which involves a mild electric shock that the animal comes to fear. We've also done a paw incision, like you had surgery on your foot, and both of those produce this vulnerability. So it seems like both psychological stressors and physical stressors can do it. You're just a little bit more vulnerable after that neonatal pain experience. And then we get into the neurobiology a little bit. So we've been studying this neuropeptide called corticotropin releasing factor, CRF, we call it. It's kind of famous for its role in the stress response, and there it's found in the hypothalamus. We're studying it in this other brain structure called the amygdala. There's a small number of cells that express it in the amygdala. Those cells develop really early. Most of the brain doesn't respond to pain or stress in kind of these first weeks of life. But for some reason, these cells do. They respond pretty highly to early life pain and stress. and then they change. They become more responsive in animals that had early life pain, although fewer in number. The receptors that they connect to change in their conformation. There's two types, one called CRF receptor 1, the other called CRF receptor 2. In most animals, you get more CRF receptor 1 than 2. When these animals have neonatal pain, it's the opposite. You have more CRF receptor 2 than 1. And so we're really kind of honing in on what's changing as a result of this neonatal pain. And as far as I can tell, we're the only ones studying that. So these are discoveries that my students are making.
Inducing pain causes the amygdala to change, and then it stays changed?
That's exactly right. And that's what we think is happening with this second stressor. We think that this is part of our stress and pain system, these cells. The neonatal pain activates this system maybe at a higher level than it normally would. And that overactivation is causing the development to change, right? There's some, we call it compensatory mechanisms. The brain is trying to kind of maintain what we call homeostasis. It's trying to adapt to these changes and kind of, temper itself down a little bit. And that causes what we think now are lasting or maybe permanent changes in these systems.
And you said your students are working on this. And is it graduate students, undergraduate, a mix? Who is lucky enough to work with you on these projects?
Well, that's kind. I hope they think of themselves as lucky. I'm not always sure they do. Actually, I have really two passions. One is in the research question itself. I think it's a very important question. I'm honored that we're able to work on it. But my other passion is sharing science with others and kind of sparking science in the next generation. I think it's both important for my particular questions, but I think it's also important more generally. I think that our society is woefully ignorant about science, so almost purposefully so in some aspects. That's what I love about your work and the Maine Science Festival and this podcast. I think we'll all be better off when we're more scientifically literate. But that's my second great passion. And so I do, I've engaged a lot of outreach with K-12 students. And I also really enjoy teaching undergraduate, graduate, and other trainees science. So we have a robust undergraduate presence. I think UNE is a great neuroscience program for all undergraduates, but especially some who get to work in our labs, I think have an incredible experience. And so I tend to have between 5 and 7 undergraduates working in the lab in any given semester. They're good. I would put the top UNE students against the top students anywhere. I think these are good students. They're smart. They're hardworking. They're independent. And these 5 to 7 students, they come in. They spend about a semester maybe getting trained, sometimes a year, helping with other people's projects, really learning the techniques and tools we use. And then a lot of them go on and start leading projects. They take a little aspect of this and they make it theirs, right? Which I think is amazing. So, a few years ago, we were testing out whether a drug called a Fa inhibitor. Fa is an enzyme that breaks down our endocannabinoids. This is our endogenous system that cannabis works on. And these Fa inhibitors are what breaks it down. And they were saying if by inhibiting that, we could raise endogenous cannabinoid levels and reduce anxiety in our rat models. And there was an undergraduate student who was the first one in the world to know that, right? They knew the answer before anybody else did, right? Because they were the one that was injecting this experimental drug into these rats and finding out. Now, I hope I was a close second. I hope they emailed me the results pretty quickly. But they knew first. And now we're doing that same thing. We have some drugs that affect our CRF system that have undergraduate students who are injecting into rat amygdalas. And it turns out it reverses this chronic sensitivity to pain we get in our neonatal pain rats. And these students are the first ones to know, right? They're the first ones to collect the data. They're the first ones to analyze it. They email me the graphs and they let me know, look, it works. And I think that's awesome. I think that excitement and that kind of sense of discovery to be the first one in the world to know something. Nobody else knows it yet.
Do they realize that? Or do you have to tell them, like, you are the first one? I mean, how many people know that and how many are just kind of like, oh, that was interesting. Let's move on to the next thing.
Yeah, it's interesting. I try to emphasize that in the experience they're having. They talk about, I've heard a few people say, so I'll steal their words. You can really predict somebody's success in science by how long it takes them to look at their data. There are the people who need to know the answer right now. They've run the experiments and they need to know if it worked. And those people have that, innate curiosity and drive. They're going to do well in science. Every now and then you get students who you have to email. Like, didn't you run the experiment a couple of days ago? How did it turn out? Like, oh, I don't know. I haven't really looked yet. And you're like, oh, I don't know. Like, I'm worried about you. They don't have that intense drive, that intense curiosity to find out what happened. I have, kind of 5 to 7 undergraduate students at any given time. Because of the COVID, we're a little bit down now. We have to spread out people more. We can't have so many people in the lab at the same time. I then have a full-time research technician. He's super good, really helps the lab, helps schedule those students, helps make sure everything's ready to go. really talented. I have a master's student who's just finishing. I have a PhD student who's rotating through right now, and I hope she'll decide to stay. And I have a postdoctoral scholar who's actively applying for jobs. He's trying to find his first faculty position. So we have a really good group, a big group, way bigger than I originally imagined I'd ever have. But they're great. They're a great team. They're really fun. They get along, and they're doing good work.
So it seems to me one of the benefits of having such a large group is both you can answer more questions more quickly, but also everyone would bring a different set of strengths or a different way to, not completely different, but different ways to look at the same problems, and maybe, synergistically you can get to results that you might not otherwise.
I think that's true. Okay. I was talking to one of my colleagues a couple of years ago when I was more frustrated, when I was a little bit younger, and I had an experiment that didn't work out. Just for some technical reason, both probably the hypothesis was wrong, but also for some technical reasons, we just weren't able to really address it. And he made the point, given all the factors, right, all the materials that have to work just right, all the techniques that have to work just right, and how the rats' experiences have to be identical except that one thing that you're trying to test, and given all the variables of undergraduate schedules and seasons and changes and husbandry and all the things, it's not amazing that it doesn't work out. It's amazing that it ever works out. Right? Now, you ever get clean data that you can really interpret. And so we tend to have 3 or 4 different things going on at once, at least. And half of them just aren't going to work. And they're not going to work because our hypothesis was wrong. That's great. That's working. Right? We have an idea about what's supposed to happen. We get a clear answer that we're wrong. That's great. We learned a lot from that. But we get ambiguous things. It just didn't work, right? For whatever reason, the animals aren't anxious at all. The thing that always makes them anxious doesn't make them anxious that week. So we can't study their anxiety, regardless of the neonatal pain, or whatever it is, right? We just don't get an answer that's meaningful to us. And so we try to run 3 or 4 things at once so that 2 of them are working and giving us meaningful data. We then have some projects where we're just studying the behavior. We just wonder, does a surgical trauma work the same as a psychological trauma in creating susceptibility? Does inflammatory pain work the same as paw prick pain in creating, this resiliency? We start asking, we answer a lot of questions like that. We then have some what we call simple biological manipulations. We're just going to inject a drug. The drug's already developed. We know what it does. We're going to inject it. Then we have some more complicated things. We're going to then use transgenic rats, and we're going to use genetic molecular biology tools to manipulate those same cells. And that's going to give us a better answer, but it's technically more complicated, so it's going to take us a lot longer to get the answer, right? And so we just try to run all these things in parallel. So we have some projects finishing, some that are just starting, some that we know are going to work, some that are a little bit riskier, and we try and keep all those things running in parallel so that we're always making progress. We're always able to get some papers out. We have some preliminary data for the next grants. We're also kind of going after these really big, complicated, scientists would call sexy experiments, so that when they work out, we can get those done also. And then, students and people on my team kind of just navigate to the role they're best suited for. I've had some undergraduate students who are just super good at organizing, and they're telling the postdocs and graduate students what to do because they're good at it. They're like, you should be doing this at this time because that's an efficient time for you to do it. And you should be there. And they just get good at it. And then, I have other people who get really good at a particular technique, and they start doing it for everybody's experiment. And, just kind of those things just naturally flow out of I think, I give them a lot of independence, and they kind of find their roles, and it ends up working out pretty well.
Do you have students who are undergrads who continue undergrad school, and you are able to either collaborate or just kind of keep an eye on them to help that? I don't mean that in a bad way. I mean, at some point, if we do it right as scientists, they're going to be peers, right? So are you able to connect? And I assume you have some students who maybe even are really good at this and decide it's not for them. Does that all happen?
It does happen. When I was being trained, it was thought that if you didn't end up as a PhD academic, you were kind of a failure to science. And I just don't have that mentality. I hope my students find where they want to be and what brings them joy. And if that's in science, that's great. And if it's in academic science, that's great. And if it's not, that's great too. I hope that they have the tools to do what they should be doing for themselves. But yeah, somewhere around 50% of my students that come through the lab and stay for multiple years and graduate end up going on to either PhD, MD, DO, or other kind of terminal degree graduate school programs. Now, this is my 11th year at UNE, so we've just had a couple of students who've been around long enough to have gone through that process, and some of them are really successful. I have a couple of PhDs. I have a DO who's in neurology. She's really successful. Yeah, we've had some really good people come through. We do offer PhDs in my lab, and I have I have a student rotating through now, and I have some master's students. But mostly, I think people benefit from other experiences. So I like to send them out to find their passion somewhere else. And then, it's fun. It's fun to see them at conferences. It's fun to get the occasional email, or every now and then somebody will send me a paper. Hey, this is what I'm working on. Thought you might be curious. Yeah, it's really neat. It's neat to see them successful. It's fun to see them come from Maine. UNE's kind of we have a growing national reputation, I think, but certainly that's been growing over the time I've been here. And it's fun to see us kind of, our alums kind of reach that level where they're competitive for national PhD programs, and they're getting faculty jobs, and they're getting, prominent placements with their, medical fellowships. And it's neat. It's neat to see them succeed that way.
Yeah, I've thought for a long time that much like many of UMaine's programs, UNE has been this hidden gem that people are only just starting to come around to the strengths that exist there.
One of the neat things for me is, I came to UNE the same year the neuroscience program launched. And so I've really grown with the program, right? We have a thing called the Center for Excellence in the Neurosciences, which is a cross-college, research collaborative group. That started the year I came, as did our undergraduate neuroscience major. And so it's really been fun. I feel like I've grown up with UNE, right? As UNE has grown and expanded, so has my research and our reach. And I really feel kind of parallel with these initiatives. And it's been fun to kind of watch the whole thing grow and succeed and gain a reputation and quality. It's been really, really gratifying.
So are the next research steps just a continuation of what you've been working on and you said you were in phase 2 or stage 2 do you feel solid enough there that you're going to do the next step, or is it just going to be a merging of moving on?
Yeah. So Kate, you're probably aware that research is, the goal of research is to publish, but the necessary tools for that are funding. And so I think about my research in grant cycles. And we're in the last phase of 2 grants. I'm in the last year of one, and I have 1 year left on another. And so I'm actively in the grant applying cycle right now. And I'm optimistic and confident, so I'm not as stressed as I otherwise might be. But if we don't get funding, then the whole thing stops, right? And that's just the way it always works, and it's always in the back of my mind, right? That when the money's gone, so is the progress. So I'm actively applying for 2 grants now. I have deadlines in November. One of them is a first submission. The other is a resubmission for a grant that scored well the first time. These grants are to kind of expand upon and really, really hammer down our hypotheses, right? We really think that these CRF cells in the amygdala are responsible for these long-term consequences of neonatal pain. And we think that's actually only true in male rats. Females seem to have a whole different set of neural structures. They have the same effects. Their behavior is the same, but the neurobiology to get there seems to be pretty different. And actually, it was just part of a whole symposium on that question. And so we're going to start addressing that problem too, is trying to understand if that's the neurobiology that's happening in males, then what's the neurobiology of females? And how does that differ? And what are these brain structures? And if these grants get funded, then that's the question we're going to ask for the next few years. And assuming that we answer both those questions, we really hammer down what it is in males and we discover what it is in females, then we'll be ready for phase 3, which is to start intervening in those systems. And I hope we get the opportunity. I think there's nobody else asking these questions right now. I think that we've made a really clear and compelling case that these systems are involved. And so I hope we get to really address it. But ultimately, that's going to be up to our funders.
Well, I'm, like I said, super grateful for your time, but also really inspired by your work. I hadn't realized quite the deep dive into the animal model and how it really does tell us things about what's happening in human model. And like almost everything I do, I would love the education aspect of it. I mean, my husband is a professor at UMaine, and it seems like, any chance we've had to have students involved with something. I mean, obviously he has, but, like even down to when I was in labor with both my children, medical students were involved, which I'm not sure anyone, like that's one of those ones where people are like, you did what? And we're like, what? Isn't that how you're supposed to do it? So I always appreciate it when people are gung-ho about letting students get involved and learn stuff, because I think it's how you propagate and keep the inspiration going, but it's also how you get people who learn early that mistakes and failures are there, and that's how you get better.
Yeah, that's right. Science is an iterative process, Kate. You gotta stick with it, right, and keep trying. Yeah. And I'm always happy. I love talking to the public. I love getting out there. I love sharing what we do. I think what we do is important. I like talking about it. I think that's part of our mission too. The more we can educate people about early life pain and neonatal pain, there are just changes you can make in medical practice that can help also, aside from the science. And so I love getting the chance to talk about it.
Well, this is great. This will hold us over until we can all meet in person again. And I tap you yet again to be part of my festival.
I really appreciate the invitation to be here. I love the work you do, and I always love talking with you. And I'm honored that you'd invite me on your podcast.
The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard. The Discover Maine theme was composed and performed by Nick Parker.